quite a while since I've done a public lecture for CERT, but it's always a lot of fun. And as Kristen mentioned, I've been at CERT for quite a while. And when I came here, I was straight out of my doctoral work, and when I did my PhD, I worked on a tiny little grass. And I was always very, very envious of my friends who worked on plants with pretty flowers, that they could have nice, beautiful flower pictures on their posters to demonstrate their research. And I had a really, really hard time trying to come up with an attractive photo of my little grass that I was working on. People just didn't get so excited about that. And so when I saw an ad for a postdoc, and one of the options was working with orchids, I said, oh, orchids. That I can get good pictures of. And that was enough to sort of pull me in, and that actually is something that makes orchids really attractive to a lot of people. But once I started learning a lot more about biology behind the orchids, I was totally hooked. And this is, as Kristen said, I do a lot of other things as well. I work on Phragmites, I work on Spartina, I do a lot of other work and work with soils and trees, but this is my love here. So I really appreciate the chance to talk to you guys about it today. So, first of all, a little bit about orchids. 10% of the world's flowering plant species are orchids. Estimated 30,000 species. They occur on all continents except Antarctica, and they even occur on some of the islands just north of Antarctica. So they're not too far off. There are over 50 species in Maryland. But orchids are also widely threatened and endangered across the world. And part of the reason that they're so threatened and endangered, and one of the reasons that I found them so interesting, is their complex life histories that I'm going to tell you a little bit about today. Orchids also because of these complex life histories, tend to be the canary in the coal mine of the plant world. They sort of integrate everything that's going on in their environments. And so when something changes in the environment, the orchids are often the first ones to go. And that makes them really important from a biological standpoint, so that we can sort of use them as a way to monitor the environment. But we can also, thinking in a more positive way, use them as a way to know when we've got a healthy environment. Now, of the orchid species of the world, of the estimated 30,000 species, you can see that as far as orchid conservation goes, the 2016 IUCN Red List, nearly all of those 30,000 species have not yet been assessed in terms of whether they're threatened or endangered or whether they're secure. Of the ones that have been assessed, approximately 880 of them, only 1% have been assessed in terms of what their ecology is and what's going on. So there's an awful lot we don't know. Of those that have been assessed, you can see there are ones that are vulnerable, endangered, critically endangered, and there's also a reasonable chunk that are not threatened. But even of the few that have been assessed, there's a lot that are data deficient. We just don't know. And so there's a lot that we can learn from these species. Now, there are a lot of beautiful, beautiful native orchids in our area. There are about 220 species of native orchids in the US and Canada. 57% of those are threatened or endangered in some part of their range of distribution. That's kind of scary. And some of our most attractive native orchids, which I show in some of the pictures here, have been declining rapidly. And there was a paper just a year or two ago by Wes Knapp that was based on 40 years of data that had been collected in the state of Maryland. And all of these species here, and many others as well, had declined 50 to 90 or more percent in the last 40 years in the state of Maryland. And it has been totally unclear what we can do to help them. And orchid conservation has been mostly just a matter of, okay, we really don't know what to do to make these happy. We're just going to set that land aside and nobody's going to touch it. And that has not been very successful. And so what I want to tell you a little bit about today is some of the work that we are doing to take the research that we've been doing on orchid conservation and what it takes to make orchids grow, and to change this so that we can actually help these species recover and to conserve them. Now, we're certainly not the first people to be fascinated by orchids. Charles Darwin was famously fascinated by orchids. He was fascinated by their elaborate pollination, and also why it is that plants that produce so many seeds are so often rare. Now, if you've ever seen an orchid seed pod, it can be jam-packed with hundreds of thousands of seeds. And he calculated that if all the seeds grew into plants, then the great-grandchildren of a single orchid would cover the earth in one continuous green carpet. That's obviously really, really far from the truth. So why is it that that's the case? So why are orchids so difficult to grow and preserve? What is the orchid research telling us about this? And what will it take to apply this research to native orchids? And I'm going to finish up by where the North American Orchid Conservation Center fits in and how we are implementing the research that we're learning to help conserve orchids. Now, I told you at the beginning that orchids had complex life histories, they depend on other species, and they integrate what's going on in their environment. So if you start out with a nice little orchid, most of them rely on pollinators to get the pollen that they need from either other plants or from within a single plant to form the many, 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 many seeds. Now this is a single seed capsule here. All this little dust that you see is orchid seeds. They're referred to as dust seeds. They are very, very small. They don't have a lot of nutrients in them, so they don't have any energy to grow. In order to get that energy that they need to grow, they have to form associations with fungi. And you can see here that they develop from these little, somewhere in this picture there are tiny, tiny little ungerminated seeds, and they develop into these little things called protoforms that, as far as I'm concerned, look nothing like plants. They're not green, they look like little white worms. Nevertheless, those are little baby orchids. Even at this stage, they're obviously not green, they're not photosynthesizing, they don't have a lot of nutrients stored. They are still dependent on fungi in order to grow to the point where they become a mature plant. So what this means is that the orchids rely on a whole lot of different species in their environments. Now, pollination mechanisms. This is something some people know about. This is a lovely picture here of a Dracaea glyptodon. This is an Australian orchid, not an American, not a North American one. But this is just an example of some of the really cool pollination mechanisms that they have. So this is an orchid that has this really weird looking flower. It's got this thing over here that looks and smells an awful lot like a female thinid wasp. And what happens is the male thinid wasp goes, ooh, I found love. And it comes in and it grabs hold of its lovely lady and tries to fly off with her. And what happens is there's this nice little hinge right here. And he tries to fly off with her, and instead he goes bonk right up against the pollinia and the stigma. And that can get him the pollen stuck on his head, in which case he goes off, still looking for love, still unsatisfied, and hopefully lands on another orchid and tries to do the same, and then delivers the pollen to the stigma. So this is just an example of the really, really cool things that can go on with orchid pollination. And that's one of the things that orchids are so famous for, because they have these really intricate relationships that end up in these really weird flowers. But what I'm going to talk about today is stuff that you don't see. There's an awful lot going on with orchids below ground that we didn't know about that is really important. Obviously the pollinators are important for forming the seeds, but once those seeds are formed, they've got to have the right conditions to grow. And that depends on the fungi. So what do fungi do for plants? Now this is sort of taking a step back from just orchids. Now a mycorrhiza, myco meaning fungus, rhiza meaning root, is a symbiotic association between a plant root and a fungus. And you can see here, these are little pine seedlings. They've grown down here, they have roots. And all of this white here is fungus. And that is a mycorrhizal fungus. These are generally considered mutually beneficial associations, where the fungi they have these tiny, tiny little hyphae that can get into all the little nooks and crannies in the soil and access nutrients and water that the plant can't do just by its roots. The plant, however, has this lovely green part up top. It's photosynthesizing, it's getting carbon from the air, and it can give some of that carbon to the fungus. Fungus gives nutrients to the plant, everybody's happy. This is a relationship that's characteristic of most plants in most ecosystems. It's present in just about all plant families, and it's really important for the plants being able to access nutrients and water and to protect them against pathogens. However, orchids take mycorrhizae to an extreme. Not only do they produce really extreme flowers to attract pollinators, the orchids cheat their mycorrhizal fungi. They actually eat them. They lyse the fungi and they absorb the contents. The orchids are entirely dependent on fungi for all of their nutrition, at least early in life. And this early life stage may last for only a few months or for many, many years. Now, to my mind, I'm used to thinking of plants as by fungi. This, to me, turnabout is fair play. Now, orchid mycorrhizal fungi are not very exciting to look at, and that's part of the reason we know almost nothing about them. So orchid fungi tend to look an awful lot like smear of jelly on wood that may or may not become entirely invisible once it's dry. There are some that have a little bit more color to them, but they still end up being smear on wood if you see them at all. Most of the time they're just down in the soil or growing within the substrate. However, it turns out very widespread and common because they're so difficult to see, they're so difficult to identify, they are very poorly studied because you just can't tell them apart. But it turns out they're doing a lot of ecologically very different things. Some of them are forming mycorrhizae that are the nice, lovely, standard exchange of carbon for nutrients that I showed you before with trees. Some are parasites on other fungi. Some are pathogens. Some are decomposers. They're doing a lot of things out there that we just haven't understood before now. Now, I told you orchid seeds are very, very small. These seeds have no nutrients, and their germination requires often very, very specific fungi. Not just any old fungus will do. You have to have just the right one. And what you see here is you get the little powder. This is what these little seeds look like up close. And eventually, once they've formed an association with fungi and absorbed some of the nutrients, they can grow into these weird-looking protocorms. Eventually, those protocorms will start to green up, and then they will start to produce a leaf. Well, I want you to notice something about this. Most of the time, when you start growing plants from seed, the first thing that comes out is the root. There's no root on this. And they almost always produce their leaf first before they produce a root. Most plants will produce a root first. They've got to access nutrients. No, no, no. These guys are getting nutrients from their fungi that are growing into all of these cells here, and the orchid is digesting them. Now, this relationship continues once orchids do become green and photosynthetic, if they do that, and not all orchids do. And the fungi grow into the cells. This is a cross-section here of a root. If you look very closely, you can see a little outline here that is the root cell outline, and you see all this big coil here in the middle. This is all fungus. Okay, that is a packet of snack food for the orchid. Those are called pelotons. And what you find is if you have an adult orchid, if you were to look inside of the orchid's roots, you would find that they would be in many, many places, they would be jam-packed with fungus just like this. So, and most orchids then continue to get at least some of their nutrients from fungi as adults. The fungi also help the orchids tolerate stress, and that's one of the things that we've been able to identify. Now, orchid life histories, you normally take a plant life history and you have your seeds, you have your seedlings, you have your vegetative non-flowering plants, and you have your flowering plants. Well, orchids add in this protocorm stage. That was that little white worm stage that goes between the seed and the plant with a leaf. And you have an awful lot of orchids will go dormant for an extended period of time. And what that means is not just that they'll go dormant for the winter, which is sort of when the plants are sleeping, but the plants will go for a year, maybe even as many as 20-some years, with living below ground without producing a leaf. Now, as far as we know, they're alive and growing below ground and living off of their fungus the whole time. So what you can see here is we have a lovely little fungus right here. And the seed to protocorm transition, this is totally fungus driven. Well, not totally, but very strongly fungus driven. The dormant plants are strongly fungus driven.
And then seedlings, vegetative plants, and flowering plants are also supported by those fungi to different degrees. Of course, they also rely on light, and I'll get to that a little bit later. But right now, I want to focus on the fungi. So what have we been able to find out about how fungi affect orchid performance? This is one of our rarest orchids here. This is Isotria medialoides, or small world begonia. It's sort of commonly noted as the rarest orchid east of the Mississippi. It is very, very uncommon, and we've been very fortunate to work. It is extirpated in Maryland, unfortunately, but we've been very fortunate to work on a number of populations in West Virginia and in Virginia. So we're learning a lot about this, and this is one of the species that I'll focus on today. Now, I was talking up a fellow up here in the front before the talk about how the heck do we know where these fungi are? I told you already that they're really hard to tell apart just by looking at them. Think about how when you're trying to figure out what's in the soil. There are lots of fungi in the soil. If you talk about an amount of soil, about 1/2 gram, that's about amount of a lima bean. There are up to 150 species of fungus in that lima bean worth of soil. How do you tell which are the ones that you want? And how do you tell how much of them is there? Well, basically what we do is we extract DNA from the soil. And, it turns out also that not only are there 150 species in that lima bean part of soil, but if you take another lima bean worth of soil, from 15 centimeters away, they're only going to share 1/4 of their species. And so there's huge, huge diversity in the soil. And it's really very challenging to figure out which fungi are there. But what we do is we sequence the DNA of the fungi that are associated with our orchids, and then we design specific probes that look only at the DNA of the fungus that we are interested in. And then we can use that to figure out how much of the fungus. I'm not going to get into the details of that right now, but if you want to ask me about it later, I'll talk your ear off about it. And so what we wanted to know, we started out and we said, all right, for seed germination, how do we tell when the fungi support seed germination? We've worked out the techniques for using the DNA to determine when the fungi are where they are. Well, but is it just enough to have the fungus there? If the fungus is there, will it support seed germination, or do you have to have a lot of the fungus? So what we did was we put out little packets of seeds, and seed packets are basically, you can take either a 35 millimeter slide mount and a very, very fine mesh, put a little pinch of seeds in there, close it up, and you can see here we've got a couple of little tiny orchids growing out the top. We also have used a mesh that we can heat seal into nice little compartments. Because if you take a little dust of orchid seeds and you sprinkle it on the soil, you will never know what happened to your seeds, and you'll never find them again, because finding dust in soil just doesn't work. So if we put them in these little packets, then we can retrieve them and figure out what happened to them. So what we did was we put out these seed packets, and then we came back and we checked to see whether our seeds germinated, and then we compared that to how much of the fungus we detected in the DNA in the soil. And we did this, first of all, we did this for 3 species of orchids. And for those of you who have ever gone looking at plants out in the woods this time of year, you may very well recognize these. Well, this time of year, not all of them, but this time of year, Liparis lilifolia is just getting ready to bloom. This is the species we have here at Cirque. It's also fairly common in Maryland. Goodyear pubescens is my favorite lab rat. I think it's a beautiful plant, no matter what time of year you find it. It's also evergreen, which means that even in the dead of the winter, if I need to find an orchid to do research on, I can find this one, which is really handy. So Liparis produces a leaf in the spring. It's green through the spring and the summer. It's flowering now. It'll disperse fruits. It'll disperse seeds in the fall, like most plants this time, around this time of year. Goodyear, green all year long. Starts to flower later in the summer, disperses seeds in the fall. And then there's Tipularia, the cranefly orchid, which is kind of an oddball. So it produces a leaf in the fall. It is green through the winter. This time of the year, it has senesced, and there will be nothing left above ground until, say, August or so, when it starts to flower. So it'll flower at a time of year when it has no leaf up. It'll produce seeds then. It'll start to disperse those seeds and it'll produce the leaf for the next year. So these are three very, very different orchids in terms of what they're doing in the woods, in terms of where they grow. We wanted to say, all right, what can we tell about the fungi that tell us something about what these orchids are? And you don't need to spend much time on this diagram. Goodyear is very specific in terms of which fungi it can use. You can see these blue ones here. These came from Liparis. Liparis is probably the most specific orchid that we have ever worked with. It uses one and only one species of fungus throughout its entire range. And then there's Tipularia, which uses fungi down here, and fungi up here, and fungi just like the Liparis fungus, and fungi just like the Goodyear fungus, and some other ones that aren't even on here. So it's very, very general in terms of which fungi it can use as an adult. But it turns out it's very specific in terms of what it needs in order to germinate the seeds. So now we've identified the fungi that these different orchids need. We can go out and we can say, where are those fungi? How does it relate to seed germination? Well, the story here is more fungus means more germination. Whether I'm looking at Goodyear, Liparis, or Tipularia, the places where we got germination were those places where there was a lot more fungus. Places where we got no germination, there was a lot less fungus. Now, this seems really simple and really logical. And you look at it and you say, well, duh. But it turns out that because these fungi are so hard to identify, they're so hard to look at what they are doing in the soil, where they are, how abundant they are, this is the first time anybody's been able to show this. And being able to determine how abundant those fungi are gives us a way to measure how happy we're making our orchids, how we're improving the conditions. And that's something we've never been able to do before. And that's one of the reasons that orchid conservation has just been, well, let's just not touch that piece of land. Because how do you know if what you're doing is helping or hurting until it's too late? The other orchid that we did some work with, that was, real quick, seed germination. So we're talking just about germination. I said orchids were also very dependent on fungi as adults, and they have this stage where they can go dormant for a really long time. Now, Isotria, this is a small world pogonia again. You can see one here, this one right here, that actually has 2 flowers on it. This is a species that is very, very prone to going dormant for extended periods of time. And it's been hypothesized for a while that, well, going dormant may be just as a sign that these plants aren't that happy. But what do you do about it? And what causes it? Well, what we did was we said, let's learn something about these Isotria, about the fungi that they need, and see if we can figure out why they're going dormant. Well, it turns out that all of the fungi that Isotria relies on themselves absolutely have to have associations with trees. So they have to have the fungus, the tree, together in order to get the orchid. This is also a reason why transplanting these orchids pretty much never works. Because unless you're moving the tree with the fungus, with the orchid, you've messed it all up. And not just any old trees will work. There are only some of the trees that can host the types of mycorrhizal fungi that Isotria relies on. Beeches, oaks, hickories, birch. Those are the potential host trees for that fungus. We identified the fungi. It turns out it can use a pretty wide range of fungi. And these fungi actually form nice mushrooms that we can identify, that we can see in the woods. Unfortunately, it's a group of fungi for which there are about 200 species. And when you go to try to key them out, you see a little note that says, these are the most common 20 species of this genus. For a more extended identification, consult an expert. Because they all look pretty much the same. And the problem is that there's about 40 of them that are little red russulas that all look pretty much the same. But we were able to use the DNA sequencing to figure out what these fungi are and to look at how much of these fungi are in the soil near our orchids. And what we found was that if we look at orchids that are currently up and visible above ground, so not dormant, versus those that have been dormant for 1 year, 3 years, or areas that have no orchids, what you can see is that in 3 separate sites, the areas where the plants are up all had the most of the fungus in the soil. Those places where the plants are dormant had much less of the fungi in the soil. And those where we have never observed plants there had the least amount of fungi in the soil. So what that tells us is that the dynamics of these orchid populations are tracking very closely what's going on with the fungi. And we've actually been able to follow this forward now a year and to say, all right, those areas that had the least fungus last year were more likely to have plants that went dormant again the following year. So this tells us that these plants are really, really dependent on their fungi. So we can say, all right, some orchids need specific fungi, others may use a whole bunch of different ones. More abundant fungi are better able to support orchid seed germination and also emergence from dormancy. So they affect the plant population dynamics. If the plant has to emerge in order to flower and produce seeds that can then produce more plants. And we thought, all right, well, maybe if we can improve the conditions for the fungi, and now since we can look and see in the soil where these fungi are and how much is there, we can tell whether we're improving the conditions for the fungi. Maybe that would also improve the conditions for the orchids. So of course, we went out and did some experiments with our 3 favorite lab rats, Tipularia, Liparis, and Goodyeara. And what we did is we made these little seed packets like I showed you before, and we did an experiment where we changed conditions, and I'll show you that in a minute, but we also grew them, put in the seed packets with and without appropriate fungi. So we cultured these fungi in the lab, we added them to wood, we got them growing on the wood, and then we added a pinch of wood that's colonized with the right fungus along with our orchid seeds. So that made sure, all right, that fungus is there. If the conditions are right, that fungus should grow, right? And then we went out into 2 different forest types, 6 sites, 3 in old forests that were like 150 plus years old, such as you see here. 3 young forests in the neighborhood of 50 to 70 years old, such as you see here. You can see they're very, very different looking. This one's much more green, has a lot more going on in the understory. And then we made these little sets of 36 little rings. And in each of these rings, each of these plots, a third of these got decomposing or chipped just leaves, crushed up. A third of them got chipped tulip poplar wood. And a third of them we didn't add anything to. And half of these got the right fungi added to them, and they all got orchid seed packets. And this is a way to tell, first of all, what do we need to do to make these orchids happy, make them germinate? How do we make the fungus happy? We made sure, we said, all right, is the fungus there to begin with? Or did we need to add it? Does the fungus do better if we add wood? Does it do better if we add leaves? Or does it do better if we just leave it alone? And what I'm going to tell you about is the results from one of them, and then I'll sort of summarize the rest of them just to sort of shorten the story. But what we found is that, and this is for Goodyeara, when we added wood, we got lots more fungus, lots more of the Goodyeara fungus, and we got lots more germinating seeds that turned into protocorms. And when we added leaves, we got somewhat more fungus, not as much as wood, and a medium amount of protocorms. And if we didn't add any wood or leaves, we got very few protocorms and not very much of the right fungus. So basically what that told us is, yes, we could make the conditions better for these fungi, and that made the conditions better for the orchids. And that was the connection we wanted to be able to see. Now, I want to say also with the other 2 species, they both also tracked the abundance of the fungi. So the conditions that made the fungus happy made the orchid more likely to germinate. However, the fungi needed different things for different species. So it turned out that Liparis fungi can grow in a really wide range of conditions, but their fungi are really, really uncommon. So what had to happen is we had to put the fungi there. And they would grow in a wide range of conditions, but we had to add the fungus. It was rarely there all by itself. For Tipularia, basically it needed decomposing wood. The fungi were very widespread, but the fungus only got abundant enough to support germination when there was decomposing wood. So basically, they all followed sort of the same pattern. They all followed the fungi, and it was how do you figure out what the fungi need to make them happy, to keep the orchids happy? Now, some of the things that we're doing to sort of apply this. We are working on ways to introduce the fungi to places that you would never expect them to be. Now, this happens to be the biodiversity tree plot out at Cirque. If you're at all familiar with it, this is a tree planting where they've planted something like 18,000 tree seedlings in a bunch of different combinations of species. These were farm fields not that long ago, and so these are soils that you wouldn't necessarily expect to find these orchids in, and you certainly would not expect to find their fungi. And indeed, if you just look at the soil that you've done nothing to, there's very, very little of the right fungus in there. And we tried to add some forest soil that was from right next to orchids. And we said, all right, well, we know this soil has the right fungi in it. What happens if we add that to these biodiversity tree soils? Can we get those fungi established enough to support orchids? And indeed, that's where we get a reasonable amount of fungus established. And if we add wood chips that have been inoculated with fungus, we can also get the fungi established. Unfortunately, what's not shown on here is that even though this is somewhat successful, we've done a pretty good job of getting the fungi established, they're still only 1/10 as abundant as they are in the forest where they support orchids. So we still have some more work to do on this. But one of the things that we're doing is to try to do some work with some orchids that were part of an orchid rescue. There's a Platanthera lacera here and a Spiranthes lacera here. These were rescued from under what is now a 6,000 square foot house in western Howard County. And so these are orchids that were definitely going to be dead anyway. And that's the only time that I would ever dig up orchids and transplant them. And we're growing them in a number of places here at Cirque. We have cultured the fungi from them, and we're working to add the fungi in different ways to the places we have these planted to see if we can make the conditions right for these orchids to get growing. And what you see here, it's a little hard to see. This is a Platanthera lacera that is getting ready to bloom. It's got some nice buds right in here. And this is in the garden here at Cirque. And we are hoping that we can get some good seeds formed from this year. These are some little Spiranthes plants. They are green through the winter, and their leaves are senescing now, so they kind of look cruddy right now. And they'll flower in like October at a time when they have no leaves up. So we'll see how they're doing when it comes to be October. But they are still up and green, and we're working to sort of figure out what we need to do to make the conditions right for the fungi so that we can work on these. Now, because the fungi from all these different orchids need different things, and we don't even know what fungi different orchids need, that means that it makes it kind of difficult. You can't sort of make a blanket recommendation for how to conserve an orchid species. All of our orchids were limited by the abundance of the fungi they needed, but the fungi were all limited by different things. We added wood and leaves that increased the host fungus abundance for Goodyeara, and adding wood inoculated with orchid fungi or adding forest soil that had the fungi in it helped to increase those fungi in new soils somewhat. We're still working out these techniques. As I said, it's not all about the fungi. There's other things going on besides the fungi, and I want to just take a second to say what we've learned about the role that light plays for some species of orchids. Obviously, the stages of the orchid life cycle that are most affected by light are those that are up and green. The seedlings, the vegetative plants, and the flowering plants. Those are also important for orchid populations. Obviously, if you don't come up and you don't flower, you don't produce seeds, and there are no more seeds to form the next generation. So one of the things that we've tried, and we're going to go back to the Isotria medialoides, this rare orchid that we've done some work on. This is a plant that, as I said, is very likely to dormant and lives off of its fungi during that time. But what's going on during the time when it's up and emergent and green? One thing that had been recommended by somebody that had tried it was they went in and they said, all right, well, if we cut out some of these trees and let a little more light in. Let's see if we can get this population to do better. And they did that, and it worked great. And somebody else tried that, and their population went extinct. And actually, this is a case where learning what's going on with the fungi may very well tell us what happened. Because their fungi, as I said, are dependent on particular types of tree species in order to grow. So very likely what happened in the cases where they didn't work out well was they took out the trees that were hosting the fungus that the orchid needed. And so we decided to try a slightly more laid-back approach, and we decided to try some canopy thinning. We didn't so much take out trees, and we certainly, if we took out any small trees, we made sure they were not ones that could be supporting the orchids. And so we opened things up a little bit and let a little bit more light in to see how these orchids did. And we have, in this case, we have two different populations. One population at Prince William Forest Park, shown in yellow, and one at Fort A.P. Hill, shown in green. And at Fort A.P. Hill, we thinned. We were able to go in there and thin out the canopy, but at Prince William Forest Park, we did not. And what you can see is that there's variation from year to year, but they pretty much were tracking each other until you get to the year we thinned. And then what you see is Prince William Forest Park is now down here, and Fort A.P. Hill is up here. And so this was a huge difference in terms of the number of plants that we found. But not only were there more plants, but the individual plants grew more. So an isotria is something that it produces one leaf during the year, and that leaf has 5 little leaflets on it. So it's like one little umbrella sticking up out of the ground. And what we found was that when there was more light, that little umbrella of leaves would continue to get bigger and bigger and bigger through the year. Whereas if we had very little light, that umbrella stayed the same size all through the year. And we know that larger plants are more likely to flower and more likely to produce fruit. So we were really excited to see this, where we had these bigger plants getting bigger through the year. And indeed, what we found is that if we looked at flowering in yellow or fruiting in green at Prince William Forest Park, where we did no thinning, and at Fort A.P. Hill, where we did thin, you can see that at Prince William Forest Park, there's certainly variation from year to year, but not really any consistent trend. Whereas at Fort A.P. Hill, you can see that there's, again, variation from year to year, and we have no idea what happened in 2011 with flowering. And so we were really excited about how these different things, both the fungi acting on the parts of the plant that we don't necessarily see, as well as the parts of the plant we do see, and then the light combined with that to influence the size of the plants and their flowering and fruiting. And we think these are two different approaches that we're now trying out to look at how we can improve these populations and have them continue to keep growing. Now, lots of different fungi, lots of different orchids, lots of things left to learn. And so one of the things that we are doing to try to apply what we're learning about orchids to orchid conservation is that we, at the Smithsonian and in collaboration with a number of partners and the U.S. National Botanic Garden, have formed the North American Orchid Conservation Center to basically try to understand what fungi these different orchids need, and then to work to develop the techniques that we need to apply this information. And our mission is to conserve our native orchid heritage, basically to conserve all of the native orchids in North America. We are using a multi-pronged approach where we are trying to learn the techniques that we need to propagate the orchids. And what we need to support that is we need banks of the fungi. So we need to grow and identify the fungi. We need banks of the seeds. And we need education. And we need education to help the public understand what is going on, and to also basically just learn how important these species are in terms of reflecting the health of our environment. Now, for most plants, you can see learning to propagate, you can see the seed banks, and you can see the education, but you won't see the fungal banks. And what we know for orchids is that if you don't have the fungi, the seed banks aren't going to do you a whole lot of good. Now, this was done with a number of different partners. It was originally founded with, and Dennis here in the front row is the person who has spearheaded this effort. So if you have many questions afterwards, I may direct you to him. And we've added even more partners recently. And they basically cover a number of partners in Canada, Alaska, throughout the US. We've also added partners in Hawaii. Whoops, what did I just hit? And basically the approach to this is to say, all right, we need to conserve the diversity of the fungi that orchids need. We need to identify those fungi. So we go out and we sample, this is a root here of an orchid. We can collect just the one root, leave the orchid where it belongs. We can go into the lab and in the microscope under sterile conditions, basically tease out those little balls of fungus, those pelotons. We can transfer them through several sterile washes of water so we get rid of all the extra bacteria that might have come from the outside of the root. And we can grow them up in the lab on agar. We now have the world's largest collection of orchid mycorrhizal fungi here at CERC. And we can grow them in these little tubes. And then what we can do, we can grow them up in liquid, extract DNA, sequence that DNA to identify the fungus, because you can have a room full of things that look like this and they can all be different and they're all going to look just exactly the same. It's very frustrating until you get to the DNA sequences, which are themselves quite different. Now, as I said, we currently have the world's largest collection of orchid mycorrhizal fungi, but we've still only got the tip of the iceberg in terms of what orchids in North America associate with. So as we grow this collection, we're also working to understand what techniques we need to conserve these orchids through cryopreservation, because this takes Jay, Dennis's technician, many, many months to transfer all of these fungi every 6 months so that they stay happy and don't use up their nutrients and die. And that's not something where, as we grow this collection, that's not something that's sustainable. And so we want to we're working to develop the techniques to conserve these orchids longer term without having to handle them all the time. We're also working with seed banks to conserve the orchid genetic diversity. And these are largely regional seed banks where we have regional groups that can go out and collect the seeds from the orchids, with permits, of course. We store the seeds on desiccant. They can be stored for a very long time. Of course, orchid seeds are very, very small, so you can get an awful lot of seeds in a tiny little tube. And we're also working with USDA and National Seed Collection to store the seeds in the National Seed Storage Laboratory so that we've got backups in multiple places that these seeds are stored. We're also working with botanic gardens and with our other partners and through research to understand what we need to do to propagate these orchids. I've talked to a number of people here already before the talk that said, why are these things so hard to grow? Why do people who try to grow these orchids why is it so difficult? Well, as I said, they need specific fungi, and most people don't know what fungi are, and they don't have the means to understand what those fungi are. That's what we're doing. We're working to identify those fungi. We're also working to understand what those fungi need to grow to be happy so that they'll support the orchids, so that we can generate from these seeds the little protocorms, and this is a different looking protocorm, it's just different species, grow them up to little green plants and get them established so that we can reintroduce them to the environment, and so that we can also use them as for education at botanic gardens, so that the public can understand. We have native orchids here and they're still really cool. I mean, I went to an orchid family day one year and we had all of our pretty pictures of native North American orchids, and we had the seeds and we had the fungi, and there was a fellow who came up to it for a while and he looked at it and rubbed his chin and scratched his head and said, finally he looked at me and he said, well, I mean, botanically they're orchids, but they're not really orchids, right? And I was very confused and talked to him for a while, and it turned out that as far as he was concerned, it wasn't really an orchid. So we work to have people understand that these orchids are there, they're really cool, and they tell us a lot about what's going on in our environments. We're also working to educate the public more, not just through things like Orchid Family Day, but to increase botanical knowledge and to engage citizen scientists and the public. And we developed a website called Go Orchids. This is something that you can access on your smartphone if you're out in the field, if you happen to have cell phone coverage. But basically what it is you can go into this site and you can basically say, I am at this location or in this state. These are the conditions I'm in. I see an orchid in front of me and I don't know what it is. What could it be? So you can identify, you can start with your location as a state, or if you think you know what the orchid is, you can put in the orchid name. And you can try to key it out. You can work to identify things, and you can tell it, say, well, I don't have flowers. Or, well, I got flowers, but I don't have seeds. Or I don't have leaves. What can you tell me? And basically you can say, even if you just have leaves, you can go in and say, all right, are the leaves down at the bottom? Are they on the stems? How are they arranged? And it'll limit it. It'll basically give you what are the options. Given where you are, given what you have to look at, what might this orchid be? And it'll show you pictures of all of them so you can compare them. So what you might get is something that looks like this. So these happen to be all, well, these are all different Platantheras. That's not. But basically, if you had gotten through and you said, all right, well, these are the leaves that I have. It's this big. This is where I am. What might it be? And you could look at these things and, well, you could also have it show you pictures of the leaves instead of pictures of the flowers. And if you were to click on one of these, you could get a species page. Obviously, you would not click on a Platanthera and get a Cypripedium species page, but just to give you an example, it's got some information about the species, where it grows, what sort of ecosystem it grows in, that it's native to North America, what its distribution is. So if you're looking at something that looks like you think a Cypripedium acoli leaf and you're down here in Nevada, you've either got something that somebody planted, a really, really oddball occurrence that definitely should be reported, or you're looking at a different species here, which is probably the most likely. And so there's some information about the species. There's some ways that you can distinguish species and learning about the orchids. We've also developed this project we call Orchid-fungus. And I've got a couple of different examples here. So at this point, we commissioned an artist in Netherlands to work with us to design 25 different models of orchids. And these are all native North American orchids. And so I've got a couple of examples right here. And what they have is it's basically a punch-out model. And it has some information about the orchid on it. It has some information about the North American Orchid Conservation Center on it. And it has a QR code to link you to the North American Orchid Conservation Center website or the the Go Orchids species page. And so there's some things that you can learn about it. At this point, we have gotten sponsors to produce the dye that you need to punch out, to produce the punch-out models for, is it 7 or 9 species, Dennis? 9 species. So we have currently 9 species that are printed in punch-out models. Any of them can be downloaded off of the web, but of course then you don't have the punch-out model. You have to sit there with an X-Acto knife and cut around the fringes of the orange fringed orchid, or however you intend to do that. Basically, we can take these things, we can use them in educational opportunities. So this is Ken Cameron at the University of Wisconsin, who has taken this into the classroom. He actually printed some giant-sized ones. And to encourage students to learn about the different orchids. And then it's very tactile. It's something that you can do. I've also brought a couple of examples here that are, if you want to look afterwards, that are ones that you can see. They're very nice. They're good fun. And they help people to learn. Hey, these are some cool plants that we have that grow here, that do cool things, and you can learn some information about them. So people start to think about what's going on around, see around them. And we've developed a model where basically we formed regional groups. So throughout the country, we don't want to try and have one person try to collect every orchid throughout North America. That's not ever going to happen. So we developed these regional groups, and within each region, they work to collect all of the orchid mycorrhizal fungi. At this point, we do most of the identifying of the fungi, collect the seeds, and start to develop the propagation and educational efforts. So what does it take to learn to grow these orchids? The regional groups that we have at this point, you can see them all in sort of different shaded colors: New England, Mid-Atlantic, Florida, Georgia, Arizona, New Mexico. There's Alaska. It gets to be its own regional group. And and then Hawaii and the U.S. administered Pacific Islands. And then there's the big group in the Midwest here. And then in the sort of cross-hatched areas are places where we have made contacts. They haven't yet officially formed regional groups, but there are people that we're talking with who are interested. So you can see we're starting to get pretty good coverage of the states in North America. And even though we don't have all the states covered yet, we have then the vast majority of the orchids in North America are covered by at least one of these regional groups. So we're making really good progress. So more than 50% of the US is covered with regional groups. We've secured funds to support a development coordinator to try and bring in the funds that we need to advance this effort. We received our first endowment. We're still seeking that big endowment to assure long-term success, but isn't that always the story? And the visits to the NAOC, North American Orchid Conservation Center, and Go Orchids websites have been increasing regularly. And there was this shout out from National Geographic to basically say how cool these sites are. And then this regional model and the NAOC model is expanding beyond the US already, even though our US isn't already filled yet. I mentioned that the Hawaii is also associated with an effort from the US Forest Service that includes Palau. There are 9 Western European countries who are starting up basically a European version of NAOC. Greece and Turkey and Australia are also other areas that are doing this. So it's something that's really catching on, and I think that's great news for the orchids around the world. So there's lots of optimism about where this is going. The NAOC model is based on ecological concepts. It's based on citizen science. It's robust, it's rapidly gaining acceptance with an ever-increasing number of collaborators. And the goal of collecting samples from all of the species in the US and Canada in the next 5 years is within reach. That's something that we're making really good progress on. We think it can be done. This is also something that the NAOC concept is reaching out globally, and it's really something that's easily adapted to any different environment or country. Now, of course, like all efforts of this sort, long-term success depends on the development of a sustainable funding base, but we're making great progress. Things are happening. We're learning things about orchids. We're working to implement those things, and we think there's a lot that is going to happen in the next couple of years. Acknowledgments, there's a lot of them. I mentioned Dennis Wiggum here from CERC, who spearheaded the NAOC effort and a lot of the orchid mycorrhizal research that we do. Jay O'Neill is his technician and has been with NAOC from the start and does a lot of the running of NAOC and cultures the fungi and on and on and on. There are also other Smithsonian collaborators at Smithsonian Gardens, folks from the US Botanic Garden who have helped with both funding as well as concepts with NAOC. Hanna Rasmussen was one of the original orchid mycorrhizal fungus researchers here at CERC. We've had funding, we've been very lucky to get funding from a number of different sources. You'll see some things in here that you might not have expected as sources of funding for orchid research. And if you have any questions, you can ask me. There's my email if you'd rather just email me. You can go to the NorthAmericanOrchidCenter.org. That's the North American Orchid Conservation Center website. And as I said, Dennis is up here as well. So I'll leave you with some pretty orchid pictures, and I believe we have a couple minutes for questions.